Hi, welcome to Moreauknows. Today we're going to be learning about the Biology Essential Standard 1.1.3. We're going to be able to identify stem cells as unspecialized cells that continually reproduce themselves and have, under appropriate conditions, the ability to differentiate into one or more types of specialized cells. So this is the first part of this standard, and we're going to learn about the stem cell. Next we'll be learning about the specialized cell. But we need to find out what stem cells mean when they are undifferentiated. We also need to learn how cells become specialized and how cells differentiate into one or more type of cells. And so for our first thing that we're going to need to look at is our definition for stem cell. A stem cell is a cell that has the ability to continuously divide and differentiate or develop into various kinds of cell tissues like muscle tissues or nervous tissue or skin tissue. The characteristics of stem cells is that they're kind of like blank cells. They're unspecialized. They have the capability of dividing and renewing themselves for long periods of time. They're actually in us all the way up until we die so that we can still make new cells. So they have the potential to give rise to specialized cells, and that's what really differentiation really is. Ooh, I like the white. So what is a stem cell? Well, first thing that we need to know is how the cells are going to divide. Well, if a stem cell divides, it turns into two new identical stem cells. And that's just self-renewing. So that's making a copy of a cell, which we'll get into the more specifics of cell replication later. And the second part is making a stem cell into a specialized cell, and that's called differentiation. So we're making a cell specialized. Today we're going to be learning about three different kinds of stem cells: totipotent, pluripotent, and multipotent stem cells. The totipotent stem cell can develop into an entire new individual. So this happens when you have twins, the identical twins. The cell actually splits and separates, and you'll get an individual and another individual that will grow up, and you'll have identical twins. We can use that to gather stem cells at the beginning to create a new individual. Pluripotent cells can make any other type of specialized cell, and those are the stem cells that we want. The only problem is that they're in an embryo, in what's called a blastocyst. Multipotent cells are differentiated but can still form other tissues. So if you had a muscle cell, a multipotent muscle cell, you'd have it could turn into a cardiac muscle for your heart, or a smooth muscle for your stomach, and many other different types of cells under those categories. Stem cells are found when we have a sperm and an egg that come together. This is called a zygote. Well, when that cell comes together, it'll grow and then divide into two cells. Okay, and then those two cells will divide and grow into four. And then we'll start getting more cells, 16, and then many other cells. And so we call this a blastocyst, and this is a very early embryo. And this is where we can find all of the embryonic stem cells. Embryonic stem cells are the most valued because they have not differentiated at all. And then eventually we will get a child, which will grow into an adult. And actually, an adult, we can get adult stem cells as well. We can also get tissue stem cells from the fetus, umbilical cord, and the placenta, and even adult tissues. So from the fertilized egg to about 3 or 4 cell divisions afterwards, we get totipotent types of stem cells. After that, with the blastocysts, we get the pluripotent cells, and these are the most valued. Also, they're the most controversial because this could potentially turn into a human. And then afterwards, once the cells have differentiated, you can actually get multipotent cells, which can turn into a bunch of different cells. So here we have a blood cell that can actually turn into a white blood cell or a red blood cell. Embryonic stem cells, they're found in a blastocyst, which is the inside cellular mass. We do not really look at the outer layer. And so we can break those out of the blastocyst and gather all of these cells, and we can put them into a fluid with nutrients and actually grow them in the lab. And we can stick these onto a heart or maybe even a scaffold that we can create, and that could actually turn into a beating heart, which they actually have done. So after we grow them in a culture, the cells can actually differentiate and turn into any type of cell that we need to. So if someone has spinal cord damage, we can create a nerve cell. If someone got burned really bad, we can create skin cells. They just created a trachea for a child that was born without one. The only problem is that we can't figure out, well, we're trying to figure out all of the different conditions that makes an embryonic stem cell grow into a skin cell. And so for each one of these specialized cells, it takes a specific type of code, if you want to say, for these embryonic stem cells to undergo and signaling to get to a fully grown, mature specialized cell. And so they're all different conditions, and so it's really hard to figure out what those are and make sure you get it correct every time. The pluripotent stem cells have the most potential to become any new cell. So we can take those out of a blastocyst and create those. The multipotent stem cells, they're a little bit limited of what they can do, but they're still useful as well, and we can pick them up. We can find the stem cells as an adult, and we don't have to worry about as much of the embryonic ethical questions that are left behind. Thank you for watching Moreauknows. Now you do.